Hello, my name is Lindsay Reynolds, and I'm the Riparian and Wetland Aim Lead with the Bureau of Land Management at the National Operations Center. The following presentation will cover field methods for annual use in Section 8 of the Field Protocol for Lentic Riparian and Wetland Systems. Why do BLM monitoring projects collect annual use data? We collect annual use data to monitor livestock, wildlife, and human use at a site. Annual use data can help determine whether current grazing is meeting criteria. It can serve as an early warning indicator for management and condition. Annual use data helps inform permit renewals, and annual use data helps meet compliance. There are 3 methods protocols included in the annual use section, which I will cover in this presentation. Section 8.1, stubble height, Section 8.2, soil alteration, and Section 8.3, riparian woody species use. The materials needed to conduct annual use sampling are the woody structure and annual use data sheet or annual use only data sheet, a clipboard and pencils or an electronic device for paperless data collection, whichever is preferred, a graduated survey rod or height measuring stick with graduations in centimeters or meters, such as an avalanche pole with clear markings, a MIM frame, which is the eye-shaped quadrat frame shown in the top middle, which is 50 centimeters long by 42 centimeters tall and typically made of half-inch PVC pipes and couplers. 3 30-meter measuring tapes and chaining pins to secure the transects. The first annual use method I will cover is the stubble height method in Section 8.1. To begin stubble height methods, you must first establish and anchor the transect. Then, step 1 of the methods is to complete the top section of the datasheet or the first page on the Survey 123 form with information about the site, the observers, and recorder. Step 2, the data are collected at 2.5 meter intervals along the transect in a 50 by 20 meter quadrat, shown by the diagram on the right. The first point for data collection is at the 0 meter mark on the transect tape. At each designated mark, place the MIM frame along the transect on the opposite side of the tape from where you are standing to form a 20 by 50 centimeter quadrat. The long edge should be parallel to the tape, and the short edge should be perpendicular to the tape in line with the designated mark. The quadrat for stubble height is half of the MIM frame closest to the tape. Stubble height may be measured on specific key species identified by the monitoring objectives of a site or project, or stubble height may be measured on any graminoid closest to the corner. Make sure to check with your project lead to determine whether stubble height will be measured on any graminoid or on specific key species. Step 3 is to locate the graminoid or key species for measurement. A 7.5 centimeter tuft closest to the upper left corner of the quadrat formed by the MIM frame and transect tape, and shown by the black circle here in the diagram. Most riparian graminoids grow tightly together, forming dense mats with little separation between individual plants. Often several rhizomatous species may be growing together. Thus, the method uses the 7.5 meter diameter tuft of all co-occurring graminoid vegetation. If graminoid vegetation or key species are sparse and part of the closest tuft is partly outside the frame, measure the whole 7.5 centimeter tuft. If graminoid vegetation or key species is not at least 7.5 centimeters in diameter, do not record stubble height for that quadrat. Mark none in the species column and mark NA in the stubble height column. Stubble height is measured for graminoid species only. If the vegetation in the quadrat is a mix of graminoids and forbs, disregard the forbs and measure graminoid leaves only. Once you've identified your tuft of graminoid vegetation, step 4 is, using the measuring rod or a separate ruler, identify the dominant or key species within the tuft, and measure the median length in centimeters of all the graminoid leaves. Median is a centermost value, as opposed to a mean. For each tuft, record the USDA plant species code or unknown code of the dominant or key species in the species column of the datasheet. If there are multiple species intermingled in the tuft, record one species that is dominant in the tuft, but measure the median leaf length for the whole multi-species tuft. Next, determine if the tuft has been grazed. Mark a yes in the grazed column if it appears that any leaves in the tuft have been grazed. Otherwise, mark no. Next, record the median leaf height in the stubble height column. Measure the leaf height only, do not measure seed stalks or culms. Determining the median leaf height will take some practice. Be sure to include all leaves within the tuft. The easiest method of doing this is to grasp the sample near the base of the leaves, stand the leaves upright, and move the hand up the leaves until about half of them fall away. Then measure the height at that location, as shown in the photos to the right. All leaves, grazed or ungrazed, are measured from the ground surface to the top of the remaining leaves. All leaves within the tuft should be lifted to determine the length. Account for very short leaves as well as tall leaves. Here's an example of how stubble height would be recorded on a datasheet. For each location on the transect, in this case the 0 meter location, the dominant graminoid species in the tuft grab is recorded, in this case Elymus canadensis, ELCA. Then it's marked yes or no whether the key species are being used. Then marked yes or no whether it's grazed. Then the median leaf height is recorded in centimeters. In this case, the median leaf height of the tuft grab of Elymus canadensis is 40 centimeters. Here's an example of what the electronic data form in Survey 123 looks like for stubble height, showing information for a tuft grab of Elymus canadensis, a stubble leaf height of 40 centimeters, and yes, it is grazed. There are several measures you can take to ensure quality data collection. These quality assurance steps for stubble height are also listed in the protocol at the end of section 8.1. Please review these steps during and after each sample event. The next annual use method in section 8.0 of the field protocol is soil alteration in section 8.2. To begin soil alteration methods, you must first establish and anchor the transect, likely the same transect that has been established for LPI, other methods, and for stubble height. Step 1, the data are collected at 2.5 meter intervals along the transect in the same quadrat as stubble height, but using both sides of the membrane this time. So, a 50 centimeter by 42 centimeter quadrat, shown by the diagram on the right. The first point for data collection is at the 0 meter mark on the transect tape. At each designated mark, place the membrane along the transect on the opposite side of the tape from where you're standing. The long edge should be parallel to the tape, and the short edge should be perpendicular to the tape, in line with the designated mark. Step 2 is to locate the observation lines within the quadrat. Soil alteration is measured in both halves of the quadrat, the entire 42 by 50 centimeter monitoring frame. Mentally project 5 lines, the 2 end bars of the frame and 3 intermediate lines spaced 12.5 centimeters apart. Mentally project those across the frame perpendicular to the transect and the center bar of the frame, as shown with the red lines in the diagram. Step 3 is to count the lines that intersect soil alteration. Look at the entire quadrat, which includes both halves of the membrane. Let's talk about what counts as soil alteration on the soil surface. These are detailed in step 3 of the field protocol. The soil surface is considered altered when there is evidence of trampling, shearing, trailing, or puddling. Trampling is hoofprints, footprints, or wheel or tread track depressions in the soil at least half an inch or 13 millimeters that exposes bare soil. The depression is measured from the top of the soil surface to the bottom of the impression. Alternatively, trampling can be identified as displaced soil moved into a pile that's at least half an inch or 13 millimeters high. Shearing is the removal of a portion of soil by ungulate hooves, leaving a smooth vertical surface and an indentation of a hoofprint at the bottom or along the sides. Trailing is when hooves, feet, or wheels and treads have repeatedly moved over the same area to create a compacted or devegetated path, even though the soil may be depressed less than half an inch or 13 millimeters. Puddling is a rut, depression, or puddle that has formed from hoofprints, footprints, or wheels or treads and has held or is able to hold water or alter surface hydrology. Finally, step 4 is record the number of lines from 0 to 5 that intersect soil alteration. Record only one occurrence of alteration, trampling, compaction, puddling, or hoof shearing per line, even if more than one occurs along the line. If a single feature intercepts more than one line, record one occurrence for each line. Count only this year's soil alteration. Trampling impacts must be obvious results of current season use. Old features tend to be non-distinct. Obvious soil alterations are defined as those that are readily observed from no closer than about 2 feet from the ground surface. In general, these are impacts that are evident without kneeling close or lying on the ground. When considering paths or trails that experience use during the current season and remain unvegetated are counted. Trails that have revegetated are not counted. Record 0 for no alteration. Do not leave the cell blank if there are no alterations. Here's an example of how soil alteration would be recorded on a datasheet, showing data recorded at the 0 meter location on the transect. First, stubble height was recorded earlier, and here 2 lines in the MIM frame were intercepted with soil alteration. So a 2 is recorded on the far right cell for that line. Here's an example of what the electronic data form in Survey 123 looks like for soil alteration, showing that 2 lines in the MIM frame were intercepted with soil alteration, so a 2 is recorded in the soil alteration line. Several measures you can take to ensure quality data collection. These quality assurance steps for soil alteration are also listed in the protocol at the end of section 8.2. Please review these steps during and after a sample event. The final annual use method in section 8 of the field protocol is riparian woody species use in section 8.3. To begin, the riparian woody species use must first establish and anchor the transect. Step 1 is riparian woody use is estimated in quadrats at 2.5 meter intervals along the transect. Data collection starts at the 0 meter mark on the transect tape. At the 0 meter mark and each 2.5 meter interval after, place the measuring rod perpendicular to the transect, extending out 1 meter on both sides of the transect, as shown in the diagram to the right. The riparian woody species use quadrat is 2 meters wide, 1 meter on each side of the tape, by the length of the sample interval, which is 2.5 meters. Use a 1 meter or 2 meter rod to define the 2 meter width of the quadrat. Riparian woody use may be measured on specific key species that are identified by the monitoring objectives of a site or project, or riparian woody use may be measured on all riparian woody species. Make sure to check with your project lead to determine whether riparian woody use will be measured on all riparian woody species or on specific key species. Step 2 is to locate the closest individual of each riparian or key woody species rooted in or overhanging the quadrat. Record the location in meters along the transect associated with the quadrat in the loc column, for example 0, 2.5, 5, etc. Since there may be multiple woody species in the quadrat, there may be multiple lines in the datasheet for each location. Riparian woody species are any riparian shrub or tree with a wetland indicator status of facultative, facultative wetland, or obligate wetland. Key species, if used, are identified by local field offices or project leads. Within the quadrat, locate the first individual of each riparian or key woody species encountered when proceeding down the transect. The individuals can be rooted in or overhanging the quadrat. If multiple individuals of the same species occur within the quadrat, only the first individual encountered, in other words, located closest to the back of the quadrat, only that first one is considered. Here in this diagram, the first individual of each riparian species is shown with a blue star. Those are the ones that would be considered for woody use. If the riparian woody plant straddles the boundary of the quadrat, so some parts are rooted both inside and outside the quadrat, evaluate the entire plant even if part of the plant is outside the quadrat. If the riparian woody plant straddles the sample interval, it's rooted into 2 adjoining quadrats, include the plant in the quadrat with the most rooted stems and estimate its browse only once. Don't estimate one plant for 2 quadrats. If needed, distinguish individual plants. This can sometimes be tricky for shrubs. The rule we use is the same as for woody structure, which is consider shrubs to be separate individuals if there's at least 0.3 meter gap in between clusters of stems. In the diagram on the right, we have several woody species in this quadrat. Estimate use on the first individual of each riparian woody species as you're moving from the 0 mark towards the end of the transect. So, in this case, there are 4 Salix goodingi, or sago, individuals. Only estimate use on the first one as you're moving down the transect, which here is colored blue. There are 2 Populus Fremontii individuals, or POFR, 1 overhanging and 1 rooted in. Estimate use on the first one, which is colored blue here. There's 1 dead Tamarisk chinensis, or TACH2. We don't estimate use on dead plants, so that one is not recorded. And there's one Utah juniper, Juwas, which is an upland plant, so we don't estimate use on it either. Those 2 are colored brown. Step 4 is, for each individual, determine the available current year's leaders. Current year's leaders are the portion of the stems of woody plants that reflect the current year's growth or that extends from the terminal bud of second year growth. They often appear as long, thin, twig-like extensions growing from terminal buds that have not yet hardened into fibrous woody material. As leaders mature, cell walls thicken and harden into coarse, woody material in the second year. Browse on second year and older branches are not considered. Next, consider which leaders on a plant are available. Available woody plants have more than 50% of the current year's leaders below 1.5 meters or 5 feet, which is considered within the reach of cattle, sheep, and deer. If evaluating horse, elk, or moose browse, consider using a higher threshold of 2.1 meters. If the first individual of a riparian woody species encountered has more than 50% of the current year's leaders above the reach of the grazing animal, the shrub is considered unavailable for grazing, and the plant is not assessed for riparian woody use. Go to the next closest plant of that species within the quadrat. If all individuals of a given riparian woody species are unavailable, mark NA in the use column. Here's an example of a browsed willow or riparian species. The rod shown in the picture is 1.5 meters high for scale. In the photo, most of those leaders are below 1.5 meters, so most of them are available. Step 5 is, after you've determined if an individual is available, then determine the woody species use class of each individual. This process is repeated for the first available individual of each riparian woody or key species encountered within the quadrat. Review the browse class descriptions in Table 12 periodically while reading the quadrats to maintain precision and accuracy. Here is Table 12 from the field protocol with the woody species use classes and descriptions. The use classes range from none, 0%, to 100% in 20% increments, and the midpoint of each class is recorded for each individual. For example, the light use class is 21 to 40% use on an individual, and the midpoint, 30, is recorded. Here are some examples of browsed willows, a riparian species. The rod shown in the picture is 1.5 meters high for scale. In the photo on the left, most of those leaders are below 1.5 meters, so most of them are available. There is some browse on the lower portion of the willow, so the rating for this individual will be slight or light. In the photo on the right, that's actually another willow in the foreground that the rod is leaning against. You can see that it's all lower than 1.5 meters, so it's all available. And most of those branches have been browsed and stripped of their leaves, so the browsing on the right individual would be heavy or severe. Here's another example of browsing on some willows. There are 2 willows shown in the photo and outlined here in green. These are low willows, both less than 1.5 meters tall, and you can see the willow on the right has not been browsed at all. But the willow on the left has been browsed quite aggressively, with almost all leaves and leaders browsed. Here's an example of how riparian woody use would be recorded on a data sheet showing data recorded at the 2.5 meter location on the transect. In this example, there are 5 woody individuals, but only 2 of them are the first of a live, available riparian species. The Ribes aureum, which is marked as a use class 10 or slight, and one of the Salix goodingi, sago, which is marked as use class 50 or moderate. You can see the riparian woody use class data are recorded on the far right cell for each riparian woody species. In this case, the Tamarix chinensis, Tatch 2, is dead, the name has been circled, so we don't record use for it. And the last individual, Apopulus fremontii pofer, has leaders above 1.5 meters and is not available. Here is an example of what the electronic data form in Survey 123 looks like for riparian woody species use, showing a sago with a use class of 10 or slight. There are several measures you can take to ensure quality data collection. These quality assurance steps for riparian woody use are also listed in the protocol at the end of section 8.3. Please review these steps during and after a sample event. The annual use methods can be collected alongside the core methods in a second pass of the transect after LPI data are collected. If annual use are being collected with core methods, they're collected at the same time as woody structure, which is also collected at 2.5 meter increments along the sample transect. Here in this diagram, you can see how, in this case, you would lay the membrane down in the woody structure quadrat, measure the stubble height and soil alteration, and then measure woody structure on any woody individual in the 2 meter by 1 meter quadrat, and use on the first riparian woody individual in the 2 meter by 2.5 meter quadrat. Then you would continue down the transect to the next 2.5 meter increment, lay the membrane down for stubble height, soil alteration, and then record woody structure and use and continue down the transect. In this way, annual use can be recorded along with the core method woody structure in a second pass of the transect. This concludes the presentation for annual use methods in section 8 of the Field Protocol for Lentic, Riparian, and Wetland Systems. Thank you for listening.